Okay. Hi, everyone. Can you all hear me? Okay. Hi. So it's my pleasure to welcome our invited speaker today, Dr. Zeynep Madak-Erdogan, who's an assistant professor at the University of Illinois Urbana-Champaign in the Department of Food Sciences and Human Nutrition. This is also where she completed her PhD in postdoctoral training, where she studied estrogen receptor biology. She started her lab there in 2014 and has been very productive and accomplished a great deal in that time. So broadly, her lab focuses on using systems biology approaches to understand how nutrients and hormones impact metabolic health and breast cancer outcomes, as well as trying to elucidate molecular mechanisms underlying more aggressive breast cancers. And her lab does a lot of really fascinating, diverse and interdisciplinary research, such as understanding mechanisms of crosstalk between ER alpha and kinase signaling and how this promotes resistance to targeted therapies, understanding the role of the gut microbiome and nutrition in regulating estrogens, as well as understanding the molecular basis for outcome disparities in breast cancer in African American women. And Dr. Madak-Erdogan is the recipient of a number of prestigious awards, including the National Center for Supercomputing Fellowship, the Women in Endocrinology Young Investigator Award from the Endocrine Society, the Mary Swartz Rose Young Investigator Award from the American Society of Nutrition. And she's also done a lot of really interesting community outreach work and seeks to engage cancer patients and advocates more in cancer research. And today she's going to tell you about multi-scale analysis of nuclear receptor signaling in cancer and metabolism. So with that, please join me in welcoming Dr. Zeynep Madak-Erdogan.
Thank you, Laura, for this nice introduction, and I'd like to thank you and Matt for hosting me. It's a great pleasure to be here, seeing friends, and also making new friends. Today, I will present data from my lab that we had been working on for almost 4 years now. My lab is really interested in this intersection between nuclear receptor biology, metabolism, and cancer. We have several projects going on, as Laura described, but today I'll talk about ER alpha and metabolism crosstalk in breast cancer with a focus on therapy resistance and breast cancer cell metabolism, and also obesity-associated breast cancer. But also we have studies that focus on metabolic syndrome associated with menopause. All right, so since, my graduate training years, I've been interested in this crosstalk between estrogen receptor signaling, mainly the nuclear estrogen receptor pathway. This is the classical mode of action for the estrogens and estrogen receptors in the presence of ligands such as 17-beta estradiol. The receptor goes to nucleus, acts as a nucleation site for recruitment of other co-regulators and would impact gene transcription events. And during my PhD and later in postdoc, we also showed that a small portion of the receptor which is present in the cytosol can activate kinase signaling pathways outside the nucleus, such as mTOR pathway or MAP kinase signaling pathways. And then through the kinases that would go into nucleus and modulation of other transcription factors, we would still see some transcriptional events that are associated with that extranuclear estrogen receptor function. And in these past 4 years, we had been trying to identify estrogens that would achieve metabolic tissue selectivity by modulating these extranuclear initiated pathways, and we have achieved it through several ways. One is use of conjugated estrogens in combination with beta-doxyphene. This is clinically used for management of postmenopausal symptoms, but also we were able to show that the presence of beta-doxyphene in this mix was able to shut down these nuclear pathways and activate these pathways, which would be beneficial in the metabolic tissues. And also, as a follow-up from my postdoctoral studies, we characterize pathway preferential estrogens, or PAPs, as well as some botanical estrogens that would work only through these extranuclear initiated pathways to achieve that metabolic tissue specificity. So that's the overall metabolism or more systemic metabolism work that we are doing, and I won't go much into detail here, but that's a leeway to the rest of my talk, because in these studies and also studies from other labs, we know that estrogen receptor is, in addition to its role in proliferation and other tumorigenicity-related phenotypes, it can regulate several metabolic genes. And this is just a review from literature, all different enzymes and different pathways that were shown to be regulated by estrogen receptor. And none of these metabolic pathways are spared. Glycolysis, TCA cycle, amino acid metabolism, free fatty acid metabolism are all impacted by the estrogen receptor and modulation of this receptor using various therapeutics available. So why multi-scale approaches? Because, we want to understand activity of a certain factor. This factor can be botanical or synthetic estrogens. It can be serum components. It might be cultivar extracts, toxicants, or carcinogens. And the first thing that we do in my lab is to develop assays to screen the biological activity of these factors based on their biology. And once we identify target factors, then we go to our omics approaches. We would occasionally perform RNA-seq, ChIP-seq, metabolomics, microbiome analysis to, select our hits, and then we would go back to our model cell culture or mouse model systems to validate these factors so that we can link our findings to the physiology. And big data analysis is a big driver in my lab. In addition to data coming from these omics technologies, we are also now utilizing electronic health records, clinical data, as well as geographical info that we analyze using machine learning techniques as well as some geospatial analysis. All right, so, and what we had done so far under these estrogens and metabolism crosstalk, I will talk about two of these projects. The first one deals with how estrogen receptor regulates metabolic pathways in the cells. So here we will talk about crosstalk between estrogen receptor alpha and a nuclear export protein, exportin-1 or XPO1. And we basically showed that endocrine resistance is driven by the crosstalk by these two proteins that would result in metabolic adaptation to limiting nutrient conditions. In the second talk, we will look at how the metabolic status affects the estrogen receptor alpha function, and it will be in the context of obesity-associated breast cancer that will deal with more of the systemic metabolism effect on how estrogen receptor works. And I mentioned this, but we also did some studies on how the body or the metabolic status of the body affects the responses to drugs, mainly drug metabolism. We showed that long-term estrogen treatment actually changed the composition of gut microbiome that would impact the availability of these estrogens and estrogen metabolites. But that's a story for another day. All right, so let's start with the first project, so therapy resistance and breast cancer cell metabolism. As I mentioned, the main players here are estrogen receptor alpha and XPO1 and exportin. And we are interested in estrogen receptor positive breast cancer because of this data. This is a paper from 2016, but a recent paper came just this past summer. Basically, for estrogen receptor positive breast cancer, we have a sustained risk of recurrence beyond 5 years. So for estrogen receptor negative cancers, we see recurrence quite early in the first 3 years. That's why in clinics, the first 3 years is a good step. But for estrogen receptor positive breast cancer, the risk is sustained and it continues to be there even after 20 years of diagnosis. And also, we know that for estrogen receptor positive breast cancer, we have a sustained risk of recurrence and less breast cancer-free or disease-free survival, as well as less overall survival beyond 5 years of diagnosis. So we still need methods to select those patients that are more likely to respond to a certain endocrine therapy, and also design combination therapies to delay or prevent endocrine resistance development. And several years ago, we showed that a particular protein called XPO1, a nuclear exportin, was overexpressed during resistant progression when the cells were grown in for hydroxytamoxifen over 100 weeks. And in that study, we showed that the main cargo for exportin was ERK5, which is a MAP kinase that would, in the absence of XPO1, would basically stay in the nucleus and localized to the transcriptional start site of genes that are regulated by estrogen receptor alpha. And in that study, we showed that basically ER alpha needed ERK5 to be there for the proper transcriptional responses to estrogens. But when XPO1 is activated, then ERK5 is transported outside the nucleus, and there are two negative things associated with that. First, ER alpha doesn't have the right partner to elicit transcriptional responses to tamoxifen. And second, now ERK5 is outside in the regions of cytosol where we see active actin remodeling, and that would contribute in this case to metastatic potential of breast cancer cells. So based on this mechanistic data, we decided to follow up the role of XPO1 and understand further how XPO1 and ER alpha crosstalk would contribute to tamoxifen resistance. And exportin 1 or XPO1 is an interesting molecule because it's already being targeted clinically in prostate cancer as well as certain leukemias. So in the presence of the cargo, XPO1 would bind to the cargo. It's exported out through nuclear pore complex and release its cargo, and the cycle would go on. And we have inhibitors, pretty effective inhibitors of this process, and one of these compounds is called selinexor, and it's orally active, generally well tolerated by the patients, overall manageable side effects, and there are not cumulative drug toxicities that have been identified. So since we had this therapeutic opportunity, we wanted to combine tamoxifen with XPO1 and see if we would improve any of the responses in tamoxifen-resistant patients. But before then, we had to show that XPO1 was a relevant target in breast cancer. We looked at XPO1 mRNA levels. We showed that it was higher in luminal B subtype of breast tumors. And also, we looked at XPO1 protein expression in 2 cohorts, one in breast tumor microarrays, and in a second cohort from UT Southwestern. In both of the cohorts, we saw increased percent XPO1 positive cells and also increased XPO1 staining intensity metastaticity in, again, luminal B subtype of tumors. And luminal B subtype is the key here because they are generally more refractive to endocrine therapies. They have higher kinase signaling, and they overexpress XPO1. In the first study where we identified XPO1, we identified also an associated XPO1 signature that mainly had nuclear transport-related proteins. The levels of the signature actually determined whether or not a woman responded to tamoxifen. Those women that had low expression of the signature did better in terms of recurrence-free survival as well as distance metastasis-free survival. But those patients that had high levels of the signature, they didn't do well on tamoxifen. That potentially suggested that XPO1 levels were an indicator for the responses to tamoxifen. Then we went ahead and tried our combination in a xenograph model, which we know is tamoxifen resistant. Actually, in this case, in the presence of tamoxifen, these tumors grow better than in vehicle-treated animals. When we treat these animals with selinexor only, the tumors regress, but after we stop injection in several weeks, these tumors come back, and when we have the combination treatment, we have the sustained tumor regression. So in following studies, we were interested in why this combination was working so well compared to the single agent treatment. The first question is whether or not selinexor and 4-hydroxytamoxifen are working synergistically, or there is just a simple addition effect. And we showed that indeed these two agents were working synergistically. So when you have them together, you had a lot more than just two agents, individual or added together. Also, we performed gene expression analysis, and we showed that in the presence of the combination, we got those genes that were regulated, that were not otherwise regulated in single agent treatments. Examples are this cluster here, where we see upregulation in combination treatment or downregulation in combination treatment. And when we looked at the function of these genes, we saw that those were mainly associated with endocrine resistance and metastasis. Those gene sets that were downregulated by the combination, they were associated with endocrine therapy resistance, that were upregulated in metastasis or luminal B type of tumors. The genes that were upregulated by the combination, they were identified in studies where these gene sets were downregulated in luminal B tumors or downregulated in metastatic tumors. So again, the gene expression studies pointed out that we were hitting those pathways that were relevant to the biology that we are interested in. Just going back here, one of the gene sets that we identified was ER alpha targets that were upregulated when AKT activation is higher. So we went ahead and checked AKT status in the presence of single agents and the combination treatment. What we found was there was this differential phosphorylation of AKT. With 4-hydroxytamoxifen, we would see increased phosphorylation at the serine 473 site, whereas with selinexor only, these AKT site at threonine 308 was activated, and it was lower when we gave the combination. And what's interesting is the patterns. We love Western blots, but we actually love it even more when we show these differences in the immunofluorescence studies. Basically, with 4-hydroxytamoxifen, we see an overall increase in serine 473 phosphorylation, and with selinexor 308 phosphorylation. But look at this pattern. Basically, those cells that were dividing were the ones that had these higher phosphorylation levels. And we are following this up now and try to understand why this differential phosphorylation matter. We were able to validate this also in our xenograph models. The TAM treatment increased overall serine 473 phosphorylation, and selinexor treatment increased the 308 phosphorylation, which was less in the combination treatments. And then we went ahead and took some more general approaches to make sure that we were not just focusing on AKT just because it's exciting, but also it was one of the major pathways that came up in our kinase arrays. And also when we look at the activity of transcription factors that were downstream of different signaling pathways. I'll just highlight the results here. So we have the differential phosphorylation events. Here we have the AKT again, and the differential regulation of the downstream transcription factors that were AKT targets. And even though we know AKT is important for survival and proliferation, we also know that it's a major regulator of metabolism, and the rest of the talk will focus on this aspect, because in our gene expression analysis, again, we found that the mitochondrial gene expression and also the glycolytic gene expression were downregulated by the combination treatment. When we performed the cell phenotype assays using the seahorse flux analysis, we showed that those cells that were treated with vehicle, they are up here, if they were treated with tamoxifen, even though their glycolytic activity would decrease, their mitochondrial activity would increase. With selinexor, we would see some dampening, but with the combination, we would get to a quiescent stage that we didn't achieve in these single treatments. What is the effect of this metabolic quiescence? We looked at downstream effects in terms of, maintaining that sustained tumor regression. And again, informed by our gene expression analysis, we found that it was the autophagy that was driven by this almost nutrient or energy deprived status. We also checked apoptosis, and we couldn't see it. This is an autophagy-driven process. We see increase in autophagy compared to what you see with rapamycin with the combination treatment, and biomarkers of autophagy are increased when we treat with the combination. And around the time when we showed that, a paper came out that showed basically inhibition of XPO1 with the selinexor or similar compounds induce autophagy in C. elegans as well as in mammalian systems. So we are hitting the right targets. All right, so up till here, we have estrogen receptor and XPO1 working together in the presence of tamoxifen. We see activation of AKT through phosphorylation at this serine 473 site that affects metabolism, but we see an increase in mitochondrial respiration, and I will come back to this. With selinexor, this time we are hitting the threonine 308 activation. We see a decrease in cell metabolism, but this is not enough to get rid of the tumor. Some of the cells survive somehow and then escape, and the tumors come back. But with the combination treatment, we are greatly reducing the respiration that would eventually induce autophagy. So we got some answers, but actually we got a lot more unanswered questions, and I listed some of them here. For example, the studies so far focused on tamoxifen resistance, but how about other endocrine resistance models? And also, how does this crosstalk regulate metabolic processes that enable survival? We still don't have the exact answer to this. And it looks like we are generating some metabolic vulnerabilities when we treat these cells with single agents. Can we target these vulnerabilities to prevent endocrine resistance? And this last question, I'll come back after I present some of the preliminary data. I mentioned other endocrine resistance models. We generated some in our lab, estrogen-deprived, tamoxifen-resistant, and now we also have palbociclib-resistant clones prednisones in combination with fulvestrant or tamoxifen. And in these models, basically, we show that when we stress these cell lines, we would get an increase in their mitochondrial and glycolytic activities. And that we think is what's happening when we treat these cells with the single agents. And this is relevant because in clinic, when a woman comes back with the recurrent disease, they will be given sequential single treatments. So basically, we are giving agents that would select for those cells that might do better and increase their metabolic potential. And those would be the ones that would survive, and this woman would come back, maybe several months later with the recurrent disease that's more aggressive now and harder to manage clinically. The other question is why the single treatment is not, providing the, basically, it doesn't kill the tumors. We think that it has to do with the shift in fuel dependency. When we look at what happens to fuel preference, we found that when we treat these cells with SXR, the dependency of these tumors to glutamine increases. What does it mean? It basically means that you need glutamine so that the mitochondrial respiration can continue. And there were several papers which show that even though, we always assume the Warburg effect and the glycolysis dependence of tumors, when you inhibit the mitochondrial respiration, the tumor cells won't proliferate. So they still need that mitochondrial respiration to continue or to survive. And in this case, when we treat with the single agent, we are basically switching the fuel preference to glutamine so that the mitochondrial respiration can continue. And also there was a decrease in the flexibility, which meant that glutamine cannot alone be used as the fuel source. You still need the other fuel sources, but for those fuel sources to be used efficiently, you need glutamine to be there. And to test this, we did an experiment where we grew the cells in limited media conditions with glutamine, low glucose or high glucose. Later, we added also free fatty acids to this. But only with the glutamine, we were able to get a proliferative effect in the presence of selinexor. We checked whether this was specific for glutamine. We tested it for all the amino acids, and this was the only one. It's really glutamine-driven. But check this out. Cysteine itself basally is doing pretty well, and this is comparable to conditions where we have the full media. It's not a limited media. So we think that there is some association between glutamine and cysteine and maybe some glutathione-dependent pathways there that might provide the survival of these cells in the limiting conditions. And I keep emphasizing limiting conditions because if you think of a tumor, the center of the tumor is pretty deprived, actually. Those are the areas where you won't be getting nutrients or oxygen as efficiently. And if the tumor is able to find, these metabolites from the environment somehow, it will survive in those conditions. The other question that we want to answer is how does glutamine contribute to survival in the presence of the single agents? And we think it has to do with some of the downstream pathways that glutamine feeds. So here we have glutamine. It goes into the mitochondria, goes through the TCA cycle, but it generates or it's used for generating aspartate or some other metabolites. And when we do metabolomics, this is what we see with the single agent treatment. There is increased production of some of these amino acids that we hit with in other assays that seem to be important for survival in those limiting conditions. And basically, we know that the glutamine use is dependent on uptake of the glutamine and conversion into glutamate because BIPTES, which is a glutaminase inhibitor, inhibits this effect. And also metformin prevents this effect, and it's due to this impact of metformin on the complex 1 of the mitochondria where we are seeing, and hopefully you are getting the idea now here that we can use some of these metabolic inhibitors to prevent resistance to single agents that might be used in the clinic. So we are working on these questions, and hopefully we will have more answers soon. But now I want to move into the second project, which deals with the effect of the metabolic status on ER-alpha function. So unfortunately in US, more than 70% of postmenopausal women are obese or overweight. And there are several studies, including Nurses' Health study, that showed that if a woman gained more than 55 pounds, her risk of breast cancer increased about 50%. And also being overweight after menopause increased ER-alpha positive breast cancer risk by about 70%. So it's now well established that obesity is a risk factor for postmenopausal hormone receptor positive breast cancer. And our question was, why does obesity increase ER-alpha positive breast cancer risk in postmenopausal women? And what's the impact of obesity on ER-alpha signaling? And our hypothesis at the beginning was the serum from obese women would contain factors that would modulate the ER-alpha signaling and increase ER-alpha positive breast cancer risk. First, we looked at the impact of the BMI, or the weight of the individual, on their normal mammary epithelial cells. So we collaborated with scientists in Common Tissue Bank, which banks the normal tissue. And we looked at the differences in mammary epithelial cells in obese versus non-obese women. And what we found was that in those women that had high BMI higher than 30, that would consider as obese, they had higher expression level of cells that had keratin 18 signal that would indicate they were getting more breast cancer stem-like properties. We also used another set from Common Tissue Bank. These are susceptible samples. So women, half of the women in this cohort, they donated blood when they were cancer-free. But unfortunately, later they developed breast cancer. And in that cohort, we also women that were age, race, and BMI matched. So we had this susceptible group, and we looked at the metabolite composition of the serum and identified certain free fatty acids as well as glycerol as the agents that were higher in blood from these susceptible women. And seeing this and being in the nutrition department, I was quickly pointed out that there might be some lipolysis going on. So here we have, for example, cancer cells, but if they need fuel to continue their activities in terms of proliferation and going on to other sites, so they would basically induce lipolysis. Just keep in mind that, you know, in the mammary gland, we have these epithelial cells that are sitting right next to these adipocytes. So there would be induction of lipolysis in that environment. And so on top of that, at obesity, the overall dysregulated adipose action, this looked like a lipolysis phenotype to us. We validated it in another cohort from Midlife Women's study. So this is a cohort that was recruited from Baltimore area. It had women, postmenopausal women, that were 2 to 3 years into menopause, that were obese or non-obese. So we were able to compare whether or not what we were seeing was associated with BMI, and it was. So those women that had higher BMI had higher levels of these particular free fatty acids. But it's not too bad. Don't despair, because in the same study we had a follow-up group that were obese at the beginning. So these are the samples from that cohort. And they later lost weight. And in that group, we were able to see reduced levels of these free fatty acids. Then we wanted to see if the serum composition made a difference in how breast cancer cells behaved. And what we did was to treat our cell lines, the usual cell lines that we use in the lab, with these serum samples. It's quite easy experiment, I would say. Basically, instead of adding serum and all the other things to the media, we just mix serum 1:3 ratio and treat our cells with that mixture. And what we saw was in ER-alpha positive breast cancer cells, if the cells were treated with the serum from obese individuals, there was a statistically significant increase in proliferation. And we wouldn't see this in ER-alpha negative cell lines. Also, we saw an increase in the cell migration and mTOR pathway activation if the serum sample was coming from the obese individual. Then we went ahead and did an RNA-seq analysis. We showed that these free fatty acids regulated common as well as different group of genes. Just to locate you, so we have palmitic acid, stearic acid, oleic acid, and also linoleic acid that will be over here that are basically in this same pathway. And they had some common as well as different gene targets. One of the gene sets that were regulated by all the groups is the PI3 kinase mTOR signaling. That's one of our favorite pathways. We were able to validate activation of the mTOR signaling and AKT when we treat our cells with the single agents. An activation of the mTOR pathway, it basically told us that we might be able to use some of the agents that we developed. I mentioned the pathway preferential estrogens that were targeting ER-alpha and mTOR signaling. We went ahead and tried these agents, and in a gene expression analysis, we were able to show that about more than 60% of all these free fatty acid regulated genes were reversed when we treat the cells with the PAPs. Other gene set groups that were affected by the PAP treatment is cell proliferation. Here we have targets that are upregulated with the free fatty acid treatment. Interestingly, only 2 of our free fatty acids regulate the cell proliferation, oleic acid and palmitic acid. We didn't see this effect with linoleic acid and stearic acid. But this proliferative effect was blocked by the PAP1 treatment. When we treat the cells with ICI or fulvestrant, so that would inhibit ER action, or RED-001, which is an mTOR inhibitor, as well as PAP, we were able to block, again, free fatty acid-induced cell proliferation. Also, this proliferation dependent on uptake of the free fatty acids to the cells. And this it happens through a transporter called CD36, which was recently shown to increase metastatic potential of breast cancer cells. So when we knocked down CD36, we were able to block this proliferative effect. Then we went ahead to our serum experiments, and we asked the question if we would see the same effects. And this is what we got, and that's quite interesting because we have cells here from obese individuals that are treated with vehicle. We treat them with tamoxifen. We don't get as much decrease in cell proliferation. Fulvestrant is doing okay, but PAP1 is doing the best. And that's kind of a different profile than what we get in standard cell culture medium. Because there, tamoxifen and fulvestrant works pretty well. We don't get as good suppression by PAP. What we think is happening is in this obese media condition, there are some rewiring going on that might make these cells more susceptible to inhibition by PAP. So that's what we saw when we looked at the metabolic pathways, fatty acid metabolism, pyruvate metabolism, citric acid cycle, and mitochondrial beta activation. Beta oxidation was affected. And we were able to validate it with the metabolomics experiment. We showed that metabolites from these pathways were upregulated by oleic acid treatment that was reversed by the PAPE. And we were able to see this effect also on the metabolic phenotype of the cells. Treatment with the free fatty acid, in this case oleic acid, increased the energetic potential of these cells that was reduced by the PAP1 treatment. How does this happen? How is ER alpha involved with this process? We did ChIP-seq analysis, and we showed that in the presence of free fatty acids, ER was driven to the chromatin, but not to your classical EREs. It was mainly going to those sites that didn't have EREs, but that had binding sites for other transcription factors that were actually playing a role in regulation of metabolism. We validated increased activity of these transcription factors using a luciferase-based assay. And when we knocked down one of these targets, in this case it was PPAR alpha, we were able to block oleic acid or palmitic acid-induced cell proliferation. This is just genes from our RNA-seq experiment that we identified as PPAR alpha targets. Again, that's validating role of this transcription factor in this process. So our working model is that in obese individuals, we have an increase in free fatty acid levels in the serum, probably due to lipolysis that's going on, and that would activate mTOR signaling and increase activity of ER. ER would be recruited to those regions of the chromatin where other transcription factors that would regulate metabolism are localized, and that would impact breast cancer cell properties. And when we target this crosstalk using a compound like PAPE1, then we can hopefully reduce this obesity-driven process in individuals. Where do we go from here? All right, so I showed you this part at the beginning. So we have the nuclear receptor biology, metabolism, and cancer. Many of it is driven by big data. But some of the data that we got from the serum studies, it got us into the health disparities. And I will share some of this data with you, which we think is pretty interesting. So if you look at the breast cancer incidence in females, we have the whites with the highest incidence of breast cancer. African Americans used to have less, but now they are catching up. If you look at the death rates, though, African Americans does worst. You know, they are up here, then we have the whites that are following them. And much of this disparity is explained by the increased instance of triple-negative breast cancer in African American women. But then we started a collaboration with a group at University of Illinois in Chicago. And they obtained the following data. So this is looking at Chicago. We have south side of Chicago that's heavily African American. And this is the death rates due to breast cancer. So there is a pretty much, almost 100% overlap in the counties that we see high breast cancer deaths. And if you look at the survival of women, stratified based on the subtype, here is the survival in estrogen receptor positive breast cancer patients. If a woman is black and coming from Southside Chicago, her chance of dying from estrogen receptor positive breast cancer is 5 times higher than a Caucasian woman. We don't have it for triple negative breast cancer. Even though you have the increased incidence, the cell doesn't kill as much. So this data made us think that there might be something going on in estrogen receptor biology in African American women. And around that time, we got our hands to a sample set cohort where we had about 1/3 of the individuals identified as African American, and we performed our metabolite analysis, and we were able to show that basically simply by this analysis we were able to differentiate between samples that are coming from African American women versus a Caucasian woman. We went back to our serum experiments, and what we got there was pretty puzzling. So if the samples are coming from a Caucasian woman, and if it is coming from an obese woman, we see our increase in the cell proliferation when we put these serum samples onto our breast cancer cells. But if the serum samples are coming from an African American woman, that's high to start with, but it doesn't go further with the obesity. The main effect that we have is on the cell migration. So obesity or the serum coming from obese individuals, it increases the cell migration potential of these cells more so than what we see if the samples are coming from a Caucasian group. And that's still association. I mean, we don't know what's driving this, what's going on. And just to be the devil's advocate, I mean, we are treating cells that are coming from Caucasian individuals. What if we got cell lines that are coming from African American individuals? Would it be different? Those are still the questions that we are trying to answer. But this was the piece of data that made us go back and think again. Here we are looking at the proliferation of the breast cancer cells and association with BMI. I already showed the effect for Caucasian individuals, that's these red lines, and African American individuals that are high to begin with. But what this graph shows is that as the income level of a woman decreases, the proliferation of the cells in the presence of the serum coming that woman increases. So you are seeing a direct effect of socioeconomic status on the biology, which is pretty impressive, apparently, to health disparities people. we didn't really appreciate this data at the beginning, but now we are finding this as a useful tool to make some of these observations in relation to race versus socioeconomic status versus other factors. And we are heading to do a more detailed analysis. We are getting serum and tumor samples from Caucasian and African American women with and without breast cancer, and we will do some of the more detailed analysis that I showed you at the beginning of my talk. I will finish by mentioning a not as science-related thing that we are doing. Lauren kind of mentioned this. Till my faculty appointment, I didn't get to interact with any survivors or patients. And I think it was one of the big things that I missed during my training. And part of it is that we didn't have patient advocates available in our community. So together with another assistant professor in my university, we started Cancer Research Advocacy Group because we realized that when we approached to survivors, they were scared to talk to us because they were intimidated by presence of scientists in the room, and we realized it was due to their perception that they didn't know enough about cancer biology. So what started as, trying to train them turned into a bigger event. Now we are funded by PCORI, Patient-Centered Outcomes Research Institute, and we have a training grant for patient advocates. And I actually heard from several students this morning your clinical translation training grant that seemed to have a big impact on them because they get to see the patients. I think it's really important to have these patient advocates in our research teams because they really give us that patient perspective. They can tell us, hey, this combination, yes, it might work in cell culture or in animals, but it wouldn't work because it has so many side effects. Hey, I don't like this treatment because it makes my life miserable and I don't understand why you keep working on this. And that really puts perspective to daily activities in your lab that are going on and makes a difference. So I urge students to hopefully go and talk to survivors if you can. Twitter is a great platform to do some of this. But being a well-established cancer center here, I am sure there are good advocacy groups here. It's really valuable to include their voice in our research as well. And with that, I will stop here. Thank people, my collaborators, Natasha Marino and Anna Maria Storniolo from Common Tissue Bank, John Katzenollenbogen and Jody Floss from UIUC. our collaborators from UT Southwestern who provided us with tumors, our industry partners from Karyopharm Therapeutics, people from National Center for Supercomputing Applications for helping us with the big data analysis, and our patient advocates who had been with us from the beginning, Jamie Holloway and Jody Shoger, who is unfortunately not with us anymore, and our funding sources, as well as people from my lab. Eylem did much of the resistance work, and also Shoham contributed to the obesity and breast cancer associated work. That I thank you for listening, and I can take more questions. Yes, Jennifer. Yes, exactly. That's how we did. Yes, so we had the matrigel invasion assays, and we would salve to the upper, area without any serum, and we would have the serum from African American or Caucasian patients in the bottom. That would be the attractant, and we use BT474 cells, which respond to serum pretty well.
So that was an invasion assay that you showed?
Correct.
Oh, so it wasn't just a migration assay?
No. Yeah. Yeah.
And were they in serum starved on one side, or just?
These assays were done in what we call treatment media. Yeah. Mm-hmm.
I was thinking about your first part of the talk. In the case of the more canonical marker of cancer, such as proliferation, there's a clear advantage of having more proliferation because it gives us simply need to divide faster. But in this case, why cancer cells would intrinsically benefit from more nuclear duplication or a more general way of saying it, nuclear trafficking activities? Why would it favor one of the two?
Because of the cargo, I would say. So I mentioned two of them. One is ERK5. In this case, having ERK5 is bad for the tumor because it makes it susceptible to tamoxifen. And if it is outside, then it's going to these areas where it can contribute to metastasis, and the cell wouldn't be responsive to tamoxifen in that case. The other one is the TFEB, the transcription factor that regulates autophagy. It's also a cargo that's transported out. So it's really dependent on the cargo, and it was shown that XPO1 targets P53 and other survival factors and modulate their nuclear or cytosolic location in different type of cancers. Yeah.
The first part of your talk was when you're treating with tamoxifen driving the cells to autophagy, to inhibit their ability to go from autophagy to cargo.
Yeah, we are doing those assays now. Yeah. We don't know yet. Yeah, no. The question is, how do you know it's autophagy, or how do you know that's the...
With combination of things of the autophagy pathway.
Mm-hmm.
What will happen if you block? There will be more resistance or sensitivity with the combination.
Yeah, we are doing those assays now. Do they survive there, right? But that was the xenograft experiment that we did, right? We stopped the injections and we want them to go for up to 4 weeks. And that's when we start seeing the selinexor only arm coming back, but not the combination arm. So it's autophagy, and there are studies that show that with autophagy only you can achieve the death of the cells now. It's not the survival autophagy in that case. Yeah.
Are you using across different subtypes of breast cancer? Are you using cell lines from different ethnic groups as well? What are you really measuring?
Yeah, so we are, for estrogen receptor positive breast cancers, we are using MCF7 and T47D and BT474s. None of them are from African American women, they are all Caucasian. And for the, let's see, yeah, that's all. And for the ER negative one, we just use MDA-MB231 so far. We are limited by the amount of serum that we get. So that's why we just kept it to the 4 cell lines. But now actually one of the students in my lab, she got several cell lines from African-American women, and she's recapitulating the same system in those cells. Yeah.
Your free fatty acid exogenous experiments where you're adding it back in, have you done any experiments to evaluate like chronic use of fatty acids versus just an acute, like you're just doing the treatment and then you're measuring, but in the patient it's going to be like a chronic exposure to that?
Correct. No, we haven't done, but it's a very good point. We haven't done any of the chronic exposure. Yeah.
So the model you had for the effect of the free fatty acids, you kind of had going through PI3K and mTOR. So is the effect of that, is that mimicked by a PIK3CA mutation, AKT mutation, any of that, or is this a totally distinct metabolic phenotype that just happens to be the same?
Yeah, we don't know the mutations, but when we use inhibitors for AKT, we can block the metabolic effect at least. And now we are looking at whether those inhibitors affect the differential expression. We haven't done any studies with the mutations, which are more relevant for tumors. Yeah. Yeah.
One more question. Yeah. Does the XPO1 go up with like patient samples with aromatase inhibitors?
Oh. I'm not sure. I should go back and check. We showed it with tamoxifen. We haven't shown it with the aromatase inhibitors. My guess is it might not. I wouldn't be surprised if it is not. Let me put it that way, because with tamoxifen we need that transcriptional activity and reliance on ER partners being there for the proper transcriptional response. But with the aromatase inhibitor, that's an estrogen-deprived
Right, but now these mutations in ER are showing up in aromatase inhibitor resistant women, and so they are still reliant on ER, but it's more specifically after.
Exactly.
So it might be interesting to look at it.
Absolutely. Yeah, that's why we are generating all these resistant models now. Obviously, the best would be to have a PDX that might have those mutations that are coming from a woman that's treated with aromatase inhibitor. Yeah.
So how are you doing your proliferation assays?
Well, I haven't showed the data here, but we are doing FACS analysis as well as just your viability assay. Since we got all these genes that are associated with proliferation, we assume that the cell cycle progression is happening with our viability assays. Just viability assay or MTSSA is a fast way of doing it. We also did studies with cells that are labeled with GFP and just looked at the cell numbers, and there also recapitulated the effects.
Yeah, because MTT assays also measure metabolism.
Exactly. That's why we did the GFP labeled cells. Yeah. All right. Thank you.